It's a great pleasure to be here. Thanks, Dr. Mitra and Dr. Rittenhouse, especially listening to a lot of beautiful talks. I'm a professor in ophthalmology. I am a co-director also in the newly created Center for Nanomedicine, where the goal is to marry engineers with clinicians and basic science researchers to speed up translation. We are located in the 6th floor of the newest research building on the Hopkins campus. Age-related macular degeneration typically affects the older population. Diseases such as cerebral palsy, autism, and other diseases affect younger people. Many of these diseases have no cure. Interestingly, neuroinflammation created by or caused by activated microglia and astrocytes play a key role in pathogenesis of not only these diseases I mentioned, but many CNS disorders. We are interested in understanding the consequences of targeted therapies for neuroinflammation, not only for just attenuating inflammation, but also for having impact way beyond inflammation. Interestingly, we are interested in systemic therapies. So in my talk, I'm talking about therapies that are administered into the system, and we are trying to see whether they can have an impact in the disease. And I'll go on details as we go. So the three talking points I want to focus on are, first I want to convince you that neuroinflammation plays a key role not only in AMD and diabetic retinopathy, but in many neurological disorders. The second point I want to say is that we have found that systemic administration of dendrimers intrinsically seem to localize them in the very cells that cause inflammation. And it is a pathology-dependent uptake. So to achieve this targeting, we do not use any targeting moieties. So in my talk, the therapeutics basically are a dendrimer that are coupled to a drug that are given systemically. So we hope that, I'll show you examples of our work from a variety of disease models. I'll start with cerebral palsy because this is, since the retina and the brain are all part of the central nervous system, we are trying to apply what we learned from the brain to apply to ocular diseases. And I'll start with this, and then I'll end in there. Since this is the last session, if you feel like stopping me in between and asking me questions or throwing tomatoes, that's fine too. Yeah. So CNS disorders are a big business. We heard many beautiful studies, slides where people showed how AMD is getting big because of the aging population. In fact, it is becoming more and more clear that neuroinflammation is a key player in autism, which is something that we are exploring, and many neurological disorders with a huge associated cost. Interestingly, for many decades, neuroscience has been focusing primarily on neurons, which are the kings or the queens of the brain. People thought that glia, which stands for glue, are basically the glue that hold neurons in place. Microglia are the immune cells, or the immune defense of the brain. So they actually are the defense ministry of the brain, if you will, and the retina. Astrocytes actually perform many important functions that regulate neurons. So in recent studies over the last 2 decades, there are even books which have come out which say if neurons are the kings or the queens, who are the people, or they are the information highway, who are the people that control the neurons? So a very important role is being increasingly recognized for microglia and astrocytes. In fact, many people kind of suggest that the root of thought are astrocytes and to some extent microglia. And people believe that for 6 decades, these cells have been neglected because of the philosophical conflict between the founding fathers of neuroscience. So we view activated microglia. So microglia under quiescent state are simply in a surveillance mode. They have a ramified morphology. But when you have a disease, they take an amoeboid morphology and they go into an activated form. Many people, or we especially, would like to view microglia not only as a problem or to shut down, but also as a therapeutic target, perhaps if we could sequester nanomaterials or drug delivery devices into these cells and hold them for a long time, we could actually then use it to have an impact that goes beyond just inflammation. So if you were to look at microglia and your neurological disorder of interest, you will find several papers, especially recently, that point out very interesting facts. In fact, inflammation is important in many diseases, because in many cases, like for example, if you look at retinitis pigmentosa or retinal degeneration, there can be 100 different genetic causes that lead to retinal degeneration or retinitis pigmentosa. But all of them, or many of them, converge in chronic neuroinflammation. So strategies that target chronic neuroinflammation may have a faster way to the patient compared to just gene therapies, where you have to do one gene at a time, for example. So we are interested in using neuroinflammation or inflammation to have an impact both in early AMD and late AMD. If you look at the pathogenesis of AMD as it happens in humans, and this is a generally accepted pathogenesis, it typically starts with chronic oxidative stress, which leads to chronic inflammation, and it leads to cell debris accumulation, drusen formation, and basically angiogenesis or neovascularization, which we typically call late AMD. So I loosely classify this as early AMD and this as late AMD. Interestingly, you can see that oxidative stress and inflammation are key aspect of the pathogenesis of AMD. As you have an increasing population or aging population, if you want to have a broader impact on the blindness, you want to be able to treat at earlier times, like when you have dry AMD. The previous speaker beautifully showed how there are methods that are becoming available to detect this disease earlier and earlier. So to have a broader impact, you need to have simpler administration methods. So we think that if you are able to achieve targeting, systemic therapies may actually be able to do especially well if the off-target effects, which is what dendrimers do, can be minimized, and I'll show you how that happens. So targeting is a big challenge, and in fact, if you look at so many ligands, there are so many antibodies, but there are not very many products that have made it to market. To try to place it in context, you could think of the size of nanoparticles to the size of a soccer ball is the same as the size of the soccer ball to Mother Earth. So injecting a drug or a nanoparticle into the system, into systemic circulation, and asking it to go to a target cell is the same of dropping a soccer ball in the Arctic Ocean and asking it to go to Baltimore. So there are many traits that need to be inbuilt into the nanoparticles that to achieve this. So what we are interested in doing is to understand how we can control or engineer, in the long-term engineer, in the short-term access and perhaps control the behavior of cells associated with inflammation. But how can you access them? Can you actually do anything to calm them and prevent the destruction that they cause in the chronic phase? More importantly, I mean, when you're talking about addressing inflammation, we have to make sure that we address the bad component of inflammation. In inflammation, there is the yin and the yang. So what you want to do is to improve the anti-inflammatory response and shut down the pro-inflammatory response. So can we actually use these cells to reprogram the microglia to make them do good things, which is what we are interested in doing. But to overcome the barriers to deliver to the brain or the retina, first you have to overcome the blood-brain or the blood-retinal barrier, which has been described in this symposium already. But the second and the more important and somewhat neglected aspect is the fact that once you escape the blood-brain barrier, which is in many cases impaired in the presence of inflammation or disease, you need to be able to move in the brain tissue. There have been many nanoparticle strategies where people showed that you could escape the impaired blood-brain barrier even with 100 nanometer particles, but that did not lead to any improvement in therapy because the escaped nanoparticle largely stayed very close to the blood vessel that it escaped from. So a second component is moving in the brain tissue, and the third component is actually finding the cell that you're interested in. This is a formidable high-risk, high-reward challenge, but an important thing that we feel like one must recognize is to look at this problem in totality. So you have to do all of these to really achieve a response. If you just focused on overcoming the blood-brain barrier, typically people think you should be lipophilic, long circulating, and small. In fact, the particles that I use are hydrophilic, short circulating, and large. So they have the exact opposite of properties of what you would typically think you would require. And the fact that neuroinflammation impairs the blood-brain barrier actually provides us an inlet into these barriers. So we use hydroxyl-terminated, nearly neutral polyamidoamine dendrimers. These dendrimers are highly non-toxic, especially because of the fact that they have a hydroxyl dendrimers. We have published on that before. And they are cleared intact relatively rapidly. So if you, for example, inject this particular dendrimer, 24 hours later, from a healthy animal, more than 97% of the dendrimer would be cleared. And so what we are interested in doing are to use the unique interaction between these dendrimers and disease pathology to try to develop therapies. Again, we don't use any ligands. So the first example I will show you about is to target neuroinflammation in cerebral palsy. This is a collaboration between my group and Sujata Kannan, who is an associate professor in anesthesiology and critical care. Just to lighten things up, she is my wife. But just to let you know, we had been married for 10 years before we started collaborating. So what we have done could be considered career suicide, but it is not a conflict of interest. So the first thing I want to show you is something really interesting. As you very well know, with Indian wives, you get one chance. If you screw up, your history. So the fact that I'm alive to talk in front of you means that we actually got some really good data. So if you were to take this particular dendrimer, the hydroxyl dendrimer, and you actually even directly inject into the brain of a healthy animal, 24 hours later, you don't find the dendrimer anywhere in the brain. The first point. However, if you so what I'm trying to say is that a healthy dendrimer not only doesn't get to the brain, but even if you put it into the brain, it's cleared rapidly. In contrast, if you inject the dendrimer systemically to an animal that has cerebral palsy, in this particular case, the cerebral palsy is created by periventricular leukomalacia, which is inflammation in the periventricular region. In this case, red is the dendrimer. And we have done all kinds of immunohistochemistry to show what those cells are and stuff like that. For want of time, I won't show you. What you see is that the dendrimer is localized in the very region where you have the problem. And it is not localized in the other region. Here, blue are the cells in the other region. If you zoom in, within 4 hours after you injected, the dendrimer appears to walk past oligodendrocytes, neurons, and other things, but they actually beautifully localize into activated microglia and astrocytes. So we found, we have tried to study, why do these dendrimers do it? Why was this not reported before? So we have done some studies. It is really difficult to prove that all the other materials don't do it. It's an expensive, unfundable, non-publishable kind of area of research. So we have to we try to get a sample of why this might be. So if you inject dextran FITC into the same animals with CP, we see that the dextran FITC stays stuck to the leaky blood vessels. If you inject 20 nanometer polystyrene nanoparticles, they also stay stuck to the leaky blood vessels. In contrast, the dendrimers actually label the cells beautifully, quickly, and rapidly. This is in an animal model. I can't tell you what it is, but you can see that in 15 minutes, it's in the parenchyma. In 4 hours, it's beautifully into the cells. So the dendrimer is able to escape the impaired blood vessel due to the disease pathology, and it is able to walk beautifully in the brain tissue. And for some reason, which I cannot disclose yet, that they find the very cells that cause the problem. So can this really be? So we have now shown that this physics happens in neonatal stroke, adult stroke, some models of autism, in a large animal, in a dog model of brain injury caused by hypothermic cardiac arrest, and so on, and in many ocular diseases. So can you really use this fact to produce a therapeutic effect? So what we did was we took this drug called N-acetylcysteine. Again, the role of inflammation and oxidative stress, not only in the problems that we are interested in, but also many CNS disorders is also described before. So NAC is a beautiful compound. It's safe. It's approved for clinical use. And it has both antioxidant and anti-inflammatory properties. We took it, we conjugated it to the dendrimer, approximately 28% by drug. And it's a highly beautifully soluble compound, 20,000 Daltons, 5 nanometers. And we have shown how we have characterized this really well. We studied how the drug comes off the dendrimer. In this particular case, the drug is designed to be cleaved by intracellular glutathione. So when the dendrimer, so outside the cell, and inside the cell, there is a 1,000-fold difference in the glutathione concentration. So when the dendrimer goes into the cell, the glutathione basically attacks this disulfide bond and releases it. Again, we have described this before. So for want of time, I'm just going to quickly show you that this approach really works. And so these are two rabbit. Sujata has characterized the rabbit model really well. So these are two rabbits born to the same mother with CP. So you can see that they are not able to walk well. They are not able to take a few steps. They don't have balance and coordination. They have hind limb stiffness. These are characteristics of CP. In fact, this is the only animal model of CP that has the phenotype of CP as seen in humans. So after we recorded it and we did neurobehavioral scoring on these animals, we gave this animal saline and this animal got dendrimer NAC. One dose after birth. Remember that many people believe that these kind of diseases cannot be cured because the problem has occurred well before you recognized it. So can you actually have an impact to make the problem be reversed is a very big challenge. So 4 days later, the animal that got saline is not looking much better. It is again barely able to move. It has hind limb stiffness that still remains the same. In contrast, the animal got dendrimer NAC did this. Remember, it's still a 5-day-old animal. So it is able to move beautifully, pretty well for a 5-day-old. And it's actually trying to take many steps and even taking hops, and it's actually trying to walk off the mat. So this is evidence of very significant motor function improvement in CP. So we have done a lot of studies to see why it happened. Why do these animals work? And we show that free drug, even if you give it at a higher dose and many doses, doesn't work. Whereas if you give dendrimer NAC, it actually shows a very big improvement. And in terms of the locomotion score, the dendrimer NAC animals looked very similar to healthy animals that got no surgical intervention. So we tried to study why is it that there have been so many studies of free NAC in animal models and humans in neurological disorders that didn't work, but why did this work? So we found that if you want to have simply an effect on the overall inflammation or oxidative stress, you could give large doses of drugs that may lead to side effects, but it has some impact on inflammation. But if you want to have functional improvements, like improvement in myelination, neurological injury, like neuronal counts and other stuff, and motor function, you have to give targeted therapy with the dendrimer. So we showed clearly that there is a very significant benefit in a very challenging indication that comes from targeted delivery to inflammation. So this tells us that neuroinflammation can be a big weapon, and when it is attenuated in an appropriate manner, it can have very significant consequences. So we have now shown similar approaches, in other animal models which we have published, but I want to focus on the eye. So I again want to show that systemic therapies can also have impact with the similar combination with the dendrimer can also have significant impact. This is collaboration with my collaborator at Wilmer Eye Institute at Hopkins, Jerry Lutty. So first thing I want to show is that we try to study, can the dendrimer, because it is able to target inflammatory cells, is it able to, how much can we accumulate in these target cells? So to cut a long story short, this is in a ischemia reperfusion model, a mouse model. What we showed is that if you inject it intravitreally, so in the healthy eye, basically the dendrimer gets cleared very quickly, but it is retained. Here red is unfortunately my thing doesn't work very well. So in the healthy eye, it's basically cleared rapidly, but whereas in the eye that is injured, the dendrimer is retained. If you were to allow me to inject 30-fold higher amount of dendrimer systemically, so if you gave 1 microgram into the eyeball, suppose you allow me to give 30 microgram systemically, I can basically achieve relatively equal uptake and retention in the posterior eye cup. So what it shows is that within a factor of 30, the extent of accumulation of a dendrimer or a potentially attached drug that you can get can be comparable even if you give it systemically. And in fact, the dendrimer, in compared to a regular delivery, it actually sits in the cells that are actually the culprits. So again, I'll be happy to go into the details. So Jerry has developed a model of a model of AMD that mimics aspects of both dry AMD and wet AMD. Again, I'll be happy to go into details if somebody is interested in it. So in early stage, you basically on day 3 or so, you have the neovascularization starts to happen, and by day 10, you have significant amount of neovascularization, and by day 21, you have like large amount of neovascularization, and then there is it starts to come back. So the interest is over a 21-day period, and it's good because over that period, we can kind of evaluate the strength of this technology. So the fact that, so,
One more minute.
Okay, so we have, this is inflammation in the human, here green is inflammation in the human CNV, and you can see that the rat model of CNV captures many aspects of the role of microglia in the inflammation. We show again that if you give systemically the dendrimer, the red is the dendrimer, green is the macrophages, and the dendrimer actually localizes one-to-one in macrophages. And I showed you, so we think that early stage therapy with NAC could have impact in dry AMD, and combination therapy with NAC and triamcinolone could have impact on late AMD. If you give the dendrimer NAC therapy on day 3, by day 10, we actually get a very significant suppression in the neovascularization, as you can see here. So you get about 80% suppression in neovascularization that is significantly better than what you can get with free drug. So it shows that if you give it in early stage, targeting attenuator inflammation can not only shut down inflammation, but it can also shut down CNV. And we show that the dendrimer therapy here, for example, the amount of macrophages in the CNV area are significantly depleted if you give DNAC compared to if you don't give any therapy. So it shows that, again, this shows that the dendrimer NAC shuts down pro-inflammatory cytokines back to the healthy animal levels, but it actually enhances the anti-inflammatory cytokines, just one shot. So it shuts down bad inflammation and seems to promote good inflammation. So we also try to see if you give it 10 days afterwards, can you cause regression in neovascularization? So here it shows that compared to day 10 therapy, if you give no therapy on day 10, this is the extent of neovascularization. If you gave DNAC plus DTA on day 10, you asked how much neovascularization you have on day 21, there is a significant reduction. You can see that from here to here, there is a significant reduction. Of course, if you don't do anything, but you just give therapy on day 10, you again get a significant suppression. So what it shows is that systemic therapies targeted to inflammation can not only have an impact on inflammation, but it can have impact on neovascularization. It is because the inflammation and oxidative stress are way above VEGF and other things when it happens to inflammation. And this is the proof that inflammation can have an impact that goes just beyond just inflammation. So when I first suggested these studies, my colleague told me, Kannan, you think just anti-inflammatory drug is going to reduce neovascularization? You are crazy. I said, yeah, we engineers are crazy, but why don't you try it? So it actually worked out really well. And this shows that it can also attenuate retinal inflammation. So I'd be happy to describe it in detail. But the beautiful thing here is that even in the retina or the choroid, the dendrimer is found only in the regions where you have a problem. In the areas around it or in the organs, the dendrimer gets cleared very quickly. And the dendrimer can be retained in these cells up to 21 days. That's the longest we have gone after you injected it. So we think that we could do targeted intracellular sustained therapy with the dendrimers. Again, it also shuts down retinal inflammation. For want of time, I'll just summarize. So first, I want to show you that inflammation may be an important emerging frontier for nanomedicine. And for a long time, nanotechnology has focused on cancer, where you want to avoid inflammation. But actually, inflammation can be an important primary target for nanomedicine. Dendrimers, PAMAM dendrimers, neutral PAMAM dendrimers have a propensity for targeting important neuroinflammatory cells after systemic administration. Now, I'm not trying to say that all the dendrimer is going to these cells. The amount of dendrimer that is found in this targeted area is less than 0.1% of what you injected, but that is sufficient to produce dramatic and unexpected efficacies in animal models. So we can do both cause regression and also suppression. So I would like to thank my students. The data I showed you were primarily done by Dr. Mishra, Siva, who is a graduate student, and Elizabeth Nanth, who just won the Burroughs Wellcome Career Faculty Award in her first year of postdoc, and funding from these areas. And I'll be happy to take questions.
Thank you. We have time for one question. Oh, we're significantly behind the schedule. Oh, go ahead. You.
Just a small question.
Sure.
Why do you think that the dendrimers, which are PAMAM, routine dendrimer, is only going into the inflamed cells, microglia, and not the normal one? What is the difference? Why it is targeting that?
So the difference that it doesn't go into healthy microglia happens only in vivo. So when you have healthy microglia, so first of all, in a healthy brain, you don't have impairment of blood-brain barrier for the dendrimer to come in. But I also told you that if I inject in the brain, the dendrimer does not go into go into healthy microglia. So we don't fully know, but we believe that when microglia are activated, they are intrinsically more phagocytic. So it is possible that there are receptors that are overexpressed that are interacting with the dendrimer in the presence of activation. But this is a very, very challenging thing to prove unequivocally, because if you throw the dendrimer into quiescent microglia, they go into quiescent microglia. Because in vitro, pretty much anything will take up the dendrimer. So what we think is happening is that either the activated microglia are tagging the dendrimer and picking it up, or that there is a receptor-mediated interaction, or that there is basically a higher phagocytic. But it cannot be just the phagocytic nature of the activated microglia, because many people would have reported that larger nanoparticles do this. So there is something special about the small and the ability of the dendrimer to move. A lot of large particles do not migrate, so people don't see them, that they go into So it's a combination of the fact that dendrimer can move.
Liposomes, dendrimers, if you put it in the blood, the macrophages attack quickly and then chew it up.
Actually,
Why is not doing it here?
So I know unequivocally that it has nothing to do with macrophage pickup, because that happens regularly. So in fact, dendrimers are so small that people they were invented in the beginning to avoid macrophage pickup. So dendrimers do not. So what I'm showing here is nothing to do with the macrophage uptake. It has to, because that is regular macrophage. Yes. This is activated macrophage pickup. So if macrophage uptake that is typically attributed to nanoparticle clearance is in play here, then dendrimers would not be picked up because they are too small to be cleared by macrophages. So we think it has to do with the size and some unique interaction with activated microglia. I know it's a long-winded explanation, but this is the best that we understand. Yeah.
Thank you very much for excellent presentation. Our